Okay, so just thanks to Nicole and Nadine first for the invitation to present something called my research journey, which I had to think a little bit about. Fortunately, I've had a rather long journey in research, and I chose this title, A 4-Decade Odyssey of Scientific Research. And I also thought using a timeline, seeing it's such a long career, landmarks in genetics and genomics. So I start here in 1980, this little kink down here. And I just thought I should give some background to why I chose science and what the questions I was interested in. So they're very simplistic questions at that time. What are genes? How is DNA packaged? And how are genes regulated? And it sort of piqued my interest reading this little novel, I'll call it a novel, The Double Helix by James Watson, who described how they discovered or came up with the idea of a double helix and what it did. But probably a better book is The Eighth Day of Creation by Freeland Judson, which really described the scientific methods. So you do science degrees, but you don't really understand how science is done. And we did have textbooks like Molecular Biology of the Gene by James Watson, 3rd edition. That was part of my undergraduate education. So then I started to understand what genes were and how they worked. But to really understand the scientific method, I always favor reading autobiographies, and this is Francis Crick, What a Mad Pursuit. So this is Watson and Crick, and that book by Crick is a suitable antidote to the double helix because it describes what really happened. Okay, so that's where I started, and I did my undergraduate and PhD at the Department of Biochemistry in the University of Adelaide from about 1981 to 1985 as a PhD student. And I did it under the supervision of Julian Wells, who was just an outstanding lecturer and really a visionary in some ways. He helped set up the Lawn Genome Conference to promote molecular biology in Australia. And he was memorialised with the Julian Wells Medal at that conference. In fact, I was awarded the medal in 2007. I was quite honoured by that. And I include this little picture down here, which is some of the lab members to try and show you the excitement of those days of making discoveries, bringing in an X-ray film, putting on a light box and trying to read what it meant. It really was an exciting time to be in molecular biology, discovering genes, sequences. This is me getting a little bit hairy, sitting on the front steps of the Darling building. But what happened in 1980 was the publication of a paper by Aaron Klug, which is the low resolution structure of histones or histone core of the nucleosome. So this is what we're really interested in, is how DNA is packaged and what are genes. So I started working on histone genes in the Wells lab. Julian had an interest in the histone genes and proteins. We used chickens as a model system because chickens have red blood cells that retain the nucleus and are locked down with a very specialized histone H5. So we were interested in histone proteins and packaging of DNA. And lastly, the really interesting thing is how do DNA binding transcription factors, sequence-specific DNA binding transcription factors, affect gene expression. So that's where I came from. And again, I returned to my PhD. What are genes? We're going to use the histone genes as an example because they were one of the first genes to be isolated for reasons I don't need to get into, but it was really because they were repeated genes. And how is DNA packaged? Well, the histones code for the histone proteins, which package DNA, and how are genes regulated by DNA binding transcription factors. So I published 3 papers from my PhD. This is the one I put most of my effort in, which is to look at the H2A, H2B genes. And what we found when we isolated the genes in lambda clones and cosmid clones is that the H2A, H2B genes are often linked. So this panel down here shows a very short region, about 320 bases between the coding regions of H2A and H2B. And gene structure implies gene function or gene regulation. And there was an element in here which was called the H2B box, which was present in front of all H2B genes. And that became a focus of our work, is trying to work out what this did. And I spent a large part of my PhD actually physically injecting DNA into Xenophus larvae eggs and functionally testing deletions of this region. And I put this up because really it was the only game in town at the time, but now everything's been replaced with cell transfection. And so when we analysed this, we found it was very important for expression. And I just put a tag here. We wanted to know the proteins that interacted with that. And that led me to my postdoc, which I'll describe shortly. So it's interesting to consider how your PhD work actually is relevant to the present day. So I put this up here in 2020, what is epigenetics? Because epigenetics is what really histone structures and chromatin structures are about. So epigenetics can be narrowly defined as changes in gene transcription through modulation of chromatin, which is not brought about changes in the DNA sequence. How's it packaged? It's the overall chromatin composition, which can index the entire genome in any given cell. And how a gene's regulated? It's by changing this packaging. And that's how you change one cell to another. And this is what I teach now. So here it is, stuff I did as a PhD is now in a book called Epigenetics from Alice. And these two pictures down here show we had the low resolution structure in 1980. Now we've got a 2.8 angstrom structure. And this is the octagon of histone proteins wrapping around a DNA helix in blue. And it's these tails that poke out that can be modified. So my whole conception of how genes are regulated is completely different to the 1980s. In some ways, I feel like I've been on a rocket ship. Things have changed so quickly. We have human genomes now. So it's just interesting to work out how or where you end up with your PhD work. But it was entirely relevant what I was doing. And this just shows by the lectures I give now. So what I did after I finished my PhD is I wrote to Jim Watson and said, I'm interested in gene transcription and gene enhancer elements, because I've worked on the immediate promoter elements of histone proteins. And he put me in contact with Winship Herr, who's shown here with a baseball cap on. And he was looking at SV40 tumor viruses at that stage and looking at the transcription factors that interact with it. So I thought it was like the perfect postdoc, exactly what I wanted to do. This is members of the Winship Herr lab during that period. We had Japanese, German, Indian postdocs, and it was really an exciting place to be. I was in formal wear in the lab, not all that often, but it wasn't unusual to go to the dinners and things at the lab. So the lab here, shown in the upper left-hand corner here, is a really beautiful campus. It holds all the summer meetings. I was very productive during my postdoc. We had like 4 genes in development papers, a letter to the editor, and a Nature paper. So what does all this data represent? This is what I did for my postdoc. I identified the DNA binding transcription factors using a technique called electrophoretic mobility shift assay, EMSA assay. EMSA assays, I purified the proteins that were binding to the 72 base pair enhancer, and I purified to homogeneity using an oligonucleotide precipitation assay. Then I characterized the binding of this protein to its sequence, and it bound to the Optimus sequence present here. So this is the theme coming in from my PhD work. I molecularly cloned the factor that was binding using a radioactive P32 assay, and then we sequenced it, and we lined up the sequence to a couple of other genes, OCT2, UNC86, and PIT1, that were being sequenced at the same time, and we came up with a POW homology domain. And as part of that, I actually mapped the DNA binding structures and found the protein I'd cloned, which was OCT1, is a bipartite DNA binding structure. So a fair amount of work, very busy during that time, but it wasn't the only thing I did. I actually had fun. So again, here's the Cold Spring Harbor lab, but the lab had a couple of boats. And this picture here just shows the Cold Spring Harbor Yacht Club, actually, and that's the picture of the lab in the back. So the yachts are on the other side of the bay. And so I lived at Davenport House, and we could go over and sail anytime. So this is the Cold Spring Harbor Sailing Club, which was anyone that wanted to volunteer to crew the boat. And this is our boat, which was the Transposon number 97. And we would compete against the local millionaires who supported the lab. And I think we were just sort of cannon fodder. We were just out there playing with them. I don't think we won too many races, but we did the best we could. It was an enormous amount of fun. And I just have to say, for every hour that you're on the boat, you've got to spend 3 hours preparing it. So it was a lot of fun, but a lot of work as well. So once I'd finished my postdoc and had a track record in molecular biology and cloning transcription factors, I returned to Australia, and I met John Maddock in Sydney, actually, in about 1988, 89. And he said he was coming to Brisbane to set up a Centre for Molecular Biology. So I returned in 1990 as an ARC QE2 fellow to set up my own lab. And we concentrated on power transcription factors, which is the class I'd identified. I moved into human pigmentation genes. That's when I started to get interested in melanocytes and melanoma, and particularly gene evolution and human genetics. So this is the group in 1990, the Centre for Molecular Biology and Biotechnology. John Maddock, I don't know if this is actually working this pointer, but there's John. Of the group here, there's a few left. Chriso's left, Kate's over there, David Hume. So we haven't completely left. There's me with a mustache now. But I have to highlight who helped me during that time and my lab. So I had some excellent honours and research assistants and PhDs, and this particular character here, Darren Smith, has been a long-term supporter of the lab. And then we had PhD students like Angus Thompson, Neil Box, Aaron Smith, and later Darren's sister, Darren Smith, helped us as well. So these people here I'll just highlight, and I can't highlight everyone, but Peter Parsons was instrumental in getting interested in melanocytes and melanoma. So Peter wanted to do a bit of molecular biology, so he had the cell biology of growing these cells, and I had the genes. George Muscat and I shared office space and lab space for almost my entire career at the CMCB and the IMB. Jenny Stowe arrived in the mid-90s and helped me with the cell biology of cells. This person here, Rob Slade, was a PhD student, had evolution on his mind, and it changed how I looked at how I should look at genes. Nick Martin and David Duffy have been some long-term collaborators and very generous with access to their biological samples, which was the twin collection out at QIMR. So this is how I progressed, and I just want to talk a bit about the biology and the strength of genetics and comparative genomics. So here I've just got the ligands, receptors, and transcription factors that are very important for melanocytes and melanoma. And the way they're identified was human genetic syndromes like the Waardenburg syndrome, just shown here in blue. And these arrows in blue show the mouse coat colors or the mouse genes that are very representative of the Waardenburg syndrome. So we could start to identify these ligands, receptors, and transcription factors by characterizing the coat colors. I wasn't involved in the coat colors, but whatever was learned in the coat colors, I applied to the human system. So here we have a case of human piebaldism, which are due to stem cell factor and KIT ligand and KIT receptor, shown in red here. Then we had the genes, like the trosinase-related protein family, shown here in green. So here we're identifying genes and giving a genetic function. Here is a gene I worked on as a class of power protein, brain2, which I'll describe in a second. We do have a mouse knockout. It doesn't really affect the pigmentation, maybe the tail a little bit. And then we have other conditions like, as I get older, graying. So we have this gene BCL2, which affects MITF function, MITF being the key regulator of melanocytes. So that's involved in graying. And lastly, something I was not initially on this slide, and that is another ligand, melanocyte stimulating hormone, or the ACIP protein, interacting with the melanocortin-1 receptor to create these different colored mice, complete black agouti or extension, which is like the red-haired mouse, and it gives the red hair phenotype in humans. Okay, so there's a lot of biology we learned from doing comparative genomics, and I'm, at the time, was still cloning genes and working out what they did. So initially for my work in the lab, we looked at the power domain and looked at its structure, looked at the genomic locus encoding, OTF1 encoding the OCT1 protein. We cloned the brain2 gene as a gene that seemed to be a power domain gene that was specific to melanocytes. And we realized it was really important because when we knocked it out, we affected tumorigenesis in mice. So the other half of my lab then branched out into this pigmentation side. So we looked at the structure of the trosinase, TRP1, TRP2 loci, and we got very interested in the melanocortin-1 receptors. There's a lot of papers here at the time, and this is MC1R, it's just a 317 amino acid G protein coupled receptor. And we first identified it as being the gene responsible for red hair in the twins out at QIMR. And then in collaboration with Nick Hayward, we looked at MC1R as a modifier of melanoma risk and also affecting the familial melanoma gene. So a lot of really good work happened at that time and very collaborative work. After being at UQ for a few years, I decided I would need a sabbatical and branch out a little bit. And I'd met this guy, Maynard Hurlin, in the early 90s. He visited Brisbane and I got talking with him. I said, can I come and do a sabbatical? He's very generous with his time. By the way, this picture of him working at the lab is a misnomer because he hardly ever worked in the lab. He directed a huge group. And so at this time, I was interested in melanoma biology, looking at the melanoblast, which is the precursor cell to the melanocyte, and looking at melanoma metastasis. And it was a really great alignment to go to Maynard's lab. So here I learned to culture melanoblast cells, which are the unpigmented precursor to the pigmented melanocytes. We also learnt, actually in my second sabbatical there in 2005, I learnt to grow spheres, and we looked at potentially phenotype switching in the melanoma spheres. So we looked at the brain-2 and MITF factors. They didn't seem to be that differently expressed in adherent cells, but when we got into spheres and also melanoma tumors, there was this dichotomy of expression. So that resulted in a very good JID paper and an oncogene paper to describe brain-2 and MITF in the spheres. So during this time, I met a lot of interesting characters. And also, well, first of all, I met my wife. My wife worked in the lab next to Maynard, and so it was sort of like an office romance, and I got married. Secondly, I met this long-haired guy here called Helmut Scheider, and he invited me out to Graz. And that's when I realized that my name actually meant what was used to describe this fizzy, very alcoholic drink. And so it's very nice, very dangerous drink. So that's where I first learned where my name came from. And also I met this guy here, Nicholas Haas, later in my visits to the Wistar. So this is just a picture taken when Maynard was actually here in about 2014. Okay, so that was my sabbatical work, and that set me up for a lot of the other work we did on this next timeline. So here is all my work as a PhD student, postdoc, early investigator, and now it's really the genomics revolution that happened. And you know, this is the difference. We were just nuts and bolts cloning to looking at genomes now. So these are the sorts of things we did. And this is a few front covers from this time. We were looking at skin color, looking at tanning. We did the red hair work in 2014 in the twins again. And we particularly got interested in eye color and evolution in the pigmentation genes. So this is me in what the 2003 to 2014. This is a representative picture of the lab at the time. And we occupied the Institute of Molecular Biology building in about 2003, and I was there till about 2014. A really lovely building to work as, you know, the premier molecular biology building on campus. And these are some of the other collaborators. Helen Leonard was very instrumental in growing melanoblasts. Two people I'll highlight here is Tony Cook and Kim Beaumont, who were responsible for some of the early work on melanoblasts, and Kim in particular on these two HNG papers describing the function, biochemical function of the melanocortin-1 receptor. And Amy there, we did all the brain-2, well, in collaboration with this guy here that you'll see again, Darren Smith, did work on the brain-2 factor. And we also looked at the genetics of eye color and described a single SNP that could be responsible for blue-brown eye color. And that work there led to a collaboration in a Nature paper looking at the derived ancestral pigmentation alleles in mesolytic humans. And here we had the strange combination of blue eyes and dark skin in a mesolytic European population. We also managed to get a cell paper on another gene that affects pigmentation, IRF4, and we did a lot of work on some of the pigmentation antigens, tyrosinase, and its association with pigmentation. Okay, after the time, about 2014, 2015, I moved, but I have to preempt that by saying it was meeting Peter Sawyer in about 2009. So this picture was taken by Helmut Scheider, who used to visit Brisbane and talk with Peter, and this is how we got together. And this is a picture of us when we were actually writing these grants. So we had a grant in 2011, 2013, and a second one in 2014, 2016 to look at the effects of genes on neogenesis. Later in 2007, in collaboration with Mitch and a few others, we got a genomics grant to look at this exome sequences. So this combination of a clinical dermatologist that knew all about Nevi and a pigmentation geneticist that knew nothing about Nevi was a good combination, because we started to be synergistic in our grants and our publications, and that led to the Centre of Research Excellence in Neogenesis in 2015. So there was a core study, and these are all the original members, and they're still here. And there was a core study to look at Nevi, but there was also these 3 different programs that radiated from this. And I was involved in the genetics and human pigmentation of neogenesis and melanoma, this program 1. So for the rest of the time, I'll actually come into the present day and start talking about what we've been doing with neogenesis. So this is the little logo we had for our CRE, and we were addressing the questions, how and why do nevi arise? And obviously it's dependent on genotype, your host phenotype, and your environment. And in Brisbane, it was UV. We have a very large amount of UV in our environment. We know skin type's important, and we're very interested in the effect of MC1R on this effect. And there's probably too much detail here, but we need to understand the biology and genetics of melanocytes. So I was involved in growing cells from nevi as well as nevocytes. So there's more to it than just the genetics. But to return to the genetics, we're very fortunate to have a Fulbright Scholar visit us in 2009. This is Elizabeth Webb, now Elizabeth Berry, and she was the one that initially set up the investigation as a pilot study. She put a poster in one of the lifts and said, please come along and we'll look at your nevi. And this morphed into the Brisbane Nevus Morphology Study, which was funded from 2011 to 2016. So the idea was to collect melanoma cases and control cases, about 1,200. We got 1,266. And we were to look at what the differences in genetics and phenotype were as risk factors for melanoma and also for nevus morphology. So in our final analysis at the 6-year point, which we're still working on, we're comparing cases and controls. We have genotype on 1,235 using the Illumina core exome, and we have 384 whole exome sequences of some very high risk cutaneous melanoma patients. It's been a very productive time. We just didn't wait till the 6-year mark. We've been publishing all the way along, resulted in some very good papers with a number of people. But today, just to keep it brief, I'm only going to concentrate on 2 of the papers. One is the genetics. This is the main Brisbane Nevus Morphology paper with David Duffy as the first author. And this is another. Sorry to interrupt, just 5 minutes, if you could, Rita, thank you. Okay, so I'm just going to describe the outcomes of these. And this is really a paper, it was that JID paper from David was first author, described the effect of nevus morphology on melanoma and nevi counts. And this just represents the Dermatology Research Centre as it is or was recently, with a few people shown here on the left, which aren't in this photo. We're very lucky to have some very talented clinicians being involved in this work. So I'll just keep a quick, I'm going to skip all the biology and just move on to MC1R. So we're very interested in looking at MC1R as a phenotype modifier and a modifier risk factor. So we have 9 common alleles of the melanocortin-1 receptor in the European population. We have 6 genotypes here. Big R is the homozygosity at the highly penetrant MC1R alleles. Represents about 70% of people with red hair. But if you're big R, little r, about 30% of those people will have red hair as well. And what we did was ask the question, we have a risk for red hair in melanoma around 4-fold, but the highest risk for melanoma is actually the number of nevi you have. So we looked at the interaction between red hair and nevi. And here are some extreme photos. These people have red hair, but no nevi. These people have red hair, but have large numbers. And we broke it down by quartile, 1, 2, 3, 4, 5, into 0 to 4 nevi, 5 to 10, 11 to 20, and 20 plus. So the question here, what is the interaction of the red hair gene and nevus genes? So here on the left, we show this quartile, 1, 2, 3, 4, 5 in people with black hair, dark brown, light brown, blonde and red. And we get a higher association when you have red hair and nevi. But it's actually a lot clearer when we look at the MC1R genotype and total nevi. Here we have the six genotypes, wild type, up to big R. So these people have dark hair and no red hair. But their odds ratio, if you have a high nevi count, is like over 5, which I said. But if you have red hair and 20 plus nevi, your odds ratio is about 25. So this is a huge effect of the MC1R genotype and Nevi together on melanoma risk. And this is published in 2009 as Duffy is the first author in a BJD paper. This then can be correlated with the absolute risk. And I'll just point out this: if you're a male and you have red hair and high Nevi count, you have a 23% chance of having melanoma in your lifetime. So this is almost as good as familial melanoma. The other thing that we did is a paper that just came out with Erin McMenamin as first author, is look at the common genes for Nevi in a common polygenic risk score. And what we find is people with multiple melanoma have a much higher risk factor. So these are 31.2% have high risk factors for the top 10%, whereas the control is just a 10%. So look, if you carry variants in the Nevis genes, you're at much higher risk of melanoma and multiple melanoma in particular. I just want to finish with genetic predisposition, saying, look, we have a Nevi pathway, high Nevi and low Nevi, and we have MC1R, but really we need to understand what's going on here. So in this case, we have some with a high number of Nevi, and they happen to be an albinism carrier. This person here has almost no Nevi, 2 Nevis, but is multiple melanoma under 40, very high risk. And is this a DNA repair defect? So we're really trying to look at the effect of the albinism genes, and we've published a couple of papers recently on the frequency of MC1R and albinism genes in melanoma and AHM. So lastly, I just want to finish on the idea of having a person's exome and looking at the rare variants, deleterious rare variants that they carry. So this is funded by the Queensland Genomics Health Alliance. In red are the number of people with melanoma in this number of rare variants that you're carrying. So if you have 8 rare variants, you're almost all multiple melanoma patients. If you have 3, you've got about a 5-fold risk of having melanoma. So this is contributing. We don't understand this yet, but we've got a selection of 500 candidate genes, and we're seeing what sort of risk they apply. So this leads into the holistic melanoma risk stratification, where we have the common polygenic risk score, we have MC1R and Nevi, and we have the rare variants to give us our holistic risk. So just to finish, I'm still working on human pigmentation, human skin and hair color. We just published a review last year. This was my current lab at the beginning of the year. We said goodbye to Casturia and to Kaz. These people here, like Darren and David, are still around helping with the analysis. This is Katie, who does all the phenotype analysis. And Aaron's been a postdoc and a collaborator for quite a while. So I'm probably out of time. I'll just acknowledge this slide has all the names. Been an excellent collaboration between Peter and myself, with David Duffy and QMR. And, I probably just have to highlight Kaz and David Duffy to do the genetics, and Katie Lee, who looks up all, is this project manager for the Brisbane Nevis Morphology Study. And I think I've sort of made it for time. Thanks for listening.
Rick, thanks very much. That was fantastic.
Thanks, Rick.
Enjoyed that. That was fantastic.
Okay.
And should we just move straight on to Mitch's talk then?
Yep.
Please.
Sorry.
Stop
Sorry,
sharing.
stop, yeah,
So Rick, we just need
sorry
to
about
stop,
that.
yeah.
Yeah, I got out of it, didn't stop. Okay.
Okay. So, everyone can see that and hear me clearly?
Yep, all good.
Yeah, great. All right. Thanks very much, Rick. That was quite a tour de force. You've been in the world for a long time, and I appreciated all the hairy photos of yourself. Quite like that. So I want to talk to you about a story that I've been working on at UQ for about 5 years. So I've been here at the Dermatology Research Centre almost 5 years to the day. So I talked about mutational profiling in benign neoplasms of the skin. So what do I mean by that? So we've performed whole exome profiling of melanocytic lesions and keratocytic lesions. So I won't be talking to you about this today. However, just to let you know, that's on the cards. So these are really interesting entities that occur when you're a little bit older. They mostly occur from sun damage, but there are also some other underlying aspects of them, which I'll talk to you about some other time. So I'm mainly going to focus today on the common acquired nevi, as well as our congenital nevi, which occur at birth. So two main stories. So firstly, we talked about the common acquired nevi, as shown there. So just very briefly, nevi are very dynamic structures, and they change over the lifetime of the person. The total number of nevi, which Rick alluded to, is one of the strongest risk predictors of melanoma. And melanomas, importantly, can actually arise adjacent to a nevus or within a nevus. And we also well know that early intervention prevents the progression of melanoma. So we're mainly focusing on this area of research here, from nevus to primary melanoma. A lot of researchers do focus on metastatic melanoma, which I have been involved in over many years as well. But not a lot of research is happening at this end of the spectrum. So this is where we fit into that. So depending on what study you look at, the melanomas can arise from a nevus, and certainly they do. And it ranges from 4 to 80%, which is quite a ridiculous range in reality. And on merit, it's around about 30%. But who really knows, to be honest? Just because you can't see your nevus doesn't mean it's not arisen, the melanoma hasn't arisen from it. Let's just say some of them don't, just to keep it interesting. So many clinicians are faced with this patients like these that have many dysplastic looking atypical nevi, but really, where is the nevus? Oh, sorry, where is the melanoma? Is it arising from these weird looking nevi, or are they coming from the skin? So it's actually coming from both, but it's very challenging to determine which is a melanoma, because nevi mimic the appearance of melanoma. But thankfully, the transformation rate of an individual nevus is actually incredibly low. And if it was the case that every nevus transformed, we'd be in dire straits. Every person would have melanoma, and of course they don't. So this is despite the fact that nevi have a common somatic mutation in the oncogenic gene called BRAF. So BRAF is commonly mutated in melanoma in about 40 to 50% of the time. In nevi, it's around about 80% on average. This is, I'll talk to you a little bit later on, different types of nevi or different dermoscopic patterns of nevi actually have different frequencies of BRAF mutation. So thankfully, melanoma, as many other cancers, is a multi-step process. Just because you have a somatic mutation in a key oncogene doesn't mean you're going to get melanoma. You need to have a lot of other steps involved, like primarily loss of tumor suppressor genes to lead to invasion and metastatic disease. So there's a renewed push around the world to look at pre-cancers. So there's a lot of people know about pre-cancers in the sense of colorectal cancer, when you find polyps in your bowel, which can be indicative of cancer forming in the future. So we're looking particularly at pre-melanomas or pre-cancers or nevi, because they're the greatest stimulator of melanoma. So as Rick mentioned just briefly as well, since 2009, Peter, Rick, and Helmut have been collecting clinical patients recruiting to their case control study. They've all had large amounts of photos taken, either using a photo finder or the Vectra imaging machine, which obviously Peter has shown you many times over the years. And many of them also have had dermoscopic patterns classified. So we were really interested in what was happening in these different dermoscopic patterns. So since 2015, since I began with a very talented then MPhil student, Jean-Marie Tan. She's now in the dermatology training program. She re-recruited patients that had specific dermoscopic patterns so we could perform molecular analysis on them. Excuse me. So out of the 600 or so patients that 1,200 patients that we knew had the dermoscopic patterns, we re-recruited around about 30 to 40 patients and had their nevus excised. So can you see the cursor there? Yep. Well, yep, I think so. So half of it was given to the pathologist for routine histopathology, which has to happen for proper diagnosis in case there actually is a melanoma, and the other half we were able to use in the laboratory. So I'm going to talk to you about 2 published studies today of this aspect of the acquired nevi, and we're also got a lot of work still coming. We're in collaboration with Brock Christensen in Dartmouth in the US. So he's an expert in methylation analysis. So it's an ongoing study which will integrate with these other data at some point. And also postdoc in my lab, Harold Oy, is looking at the RNA and microRNA sequencing to also correlate with the rest of the data. So very briefly, to start with, we looked at targeted mutation detection, and the key genes we were looking at were BRAF and NRAS. So we used an ultra-sensitive method of detection called droplet digital PCR, just shown there. And if you're not familiar with that, can detect 1 copy out of 20,000 copies in single reaction. So each reaction has 20,000 individual things going on at once. So it's a highly powerful, and I think TRI actually has one in the building. So I highly recommend you using it if you need to. So very briefly, this was published a few years ago in BJD. So we looked at the different common dermoscopic patterns of globular, reticular, which is the more flat nevi, and these non-specific ones with a peripheral rim of globules, which indicates that the nevus is actively growing. So what we found was that the globular pattern nevi were primarily BRAF mutated with a small amount of NRAS mutations. The reticular nevi were 2/3 BRAF and 1/3 NRAS, and these actively growing nevi were 100% BRAF mutated. So this really indicates that and confirms that BRAF is a very early event in nevogenesis and as well as melanogenesis. So all up, 100% of acquired nevi have are mutated for the key genes in the MAP kinase pathway. So essentially, as Peter's mentioned before, is that if it doesn't have either one of these mutations, it's not a nevus, and that's pretty much the truth. So this story prompted a editorial and commentary by Harold Kittler, which he was very interested in why the frequency of the mutations were so high. So what he thinks, and what we also believe too, is that the BRAF mutation is not sun-induced. It's actually there at a developmental stage. It's just at a subclinical level that you can't see unless the melanocytes are actually induced by the sun to create a proliferative event to actually detect the BRAF mutation. So we are looking, actively looking in the skin to see if not only BRAF but also other somatic mutations, is it truly there all the time, and is just waiting for the right insult to occur. So we actually have supportive data of this, but that's watch this space. There's much more work to be done on that. So that's targeted mutation detection. Now I'm going to move on to whole exome sequencing. So this is looking at every gene and the surrounding untranslated regions around the genes to have complete coverage of our approximately 20,000 genes in our genome. So at the time, this had never been done. Most people were focusing on melanoma, and also if they were looking in nevi, it was really just a small panel of genes. So we didn't really know what we were going to find. I was convinced there was nothing except BRAF or NRAS, but I was actually clearly pleasantly surprised to find a lot more than those 2 key genes. So we looked at 30 nevi with the matching adjacent skin. So this is really important to determine what is considered normal, what sort of normal mutations are present in the skin, so you can remove them from the nevus to look what is only present in the nevus. And to make it obviously somatic, you need the saliva, the germline DNA. So this is probably the most famous figure in genomic research. This is a collation of different mutations per megabase as it's collated across the different cancer types. Melanoma is up here, which is the most mutations per megabase, and this is based on primarily based on UV radiation causing these mutations. So this is my terrible introduction of Naevi into that spectrum there, but I think it illustrates it quite nicely that it also has a really high mutations per megabase up there of melanoma, and that's mostly because of UV. So put simply, it's more than BRAF and NRAS present in the Naevi. And these are different substitution classes that are commonly detected from somatic sequencing. C to T changes are the most frequent mutation, somatic mutation. It also can indicate that this is a high proportion of UV-induced mutations. So you can see in the Naevi, it's around about 80% C to T changes. In the adjacent skin, it's around about 40%, so about half of that. So even though there's a the same level of UV being induced, there's a different mutation spectrum. So when you compare the Naevi versus melanoma, at just the C2T type changes, the spectrum is almost identical, which indicates that they are essentially the same thing or have the same origins, and very different compared to the adjacent skin. You can see the spectrum at the C2T level is essentially a flat line. So because we know that UV is one of the strongest inducer of these mutations, and therefore the mutations that are occurred in the genes may indeed be random, and a lot of them are just random events just from the UV damage. So to try to make sense of what's happening in these data, it's important to do mutation signature analysis. So this is not just looking at the 6 substitution classes. This is looking at the surrounding nucleotides to create a pattern. So each substitution creates 16 different mutation patterns, and across the 6 substitutions, it ends up being 96 different classes. So they are shown illustrated here. That there's a lot more mutation signatures detected now, but a few years ago, Ludmila Alexandrov was one of the leaders in this field. A very talented mathematician discovered that these different types of patterns can be correlated with different exogenous and endogenous processes. So UV signature 7 is here. These two different these major peaks here can be also now split into 7A and 7B, based on which we think is based on different the UV wavelengths. So many of the other mutation signatures are related to defects in DNA repair, which I'll talk to you briefly about later as well. So just very, just in a nutshell, nevus versus adjacent skin. The skin is in the blue and the nevus is in the reddy color. They're very different. The skin has a flat profile and the Naevi has a UV signature. So UV signature 7 was dominant in the Naevi. In the skin, I won't go into what other signatures were present, but primarily aging, the process of aging itself created the most dominant signature in the skin. So just to value add the data, besides mutations, you can also look for DNA copy number events. So in cancer, as many of you will know, unbalanced events occurs where you have loss of tumor suppressor genes and gains of oncogenes, creating essentially genomic catastrophes that lead on to metastatic disease. In Naevi, it's well known that Naevi don't have a lot of copy number changes, and in most cases, depending on what type of nevus is studied, no copy number changes are detected at all. And that's what we found in these globular Naevi, which is mainly the dermal Naevi, but not many people, if ever, had ever looked at these reticular or non-specific nevi, which are the flat acquired nevi. So what we found that was these were awash with some copy number aberrations, and outwardly that appears to be malignant or like a melanoma. But to try and make sense of this data, it's really important to look at what genes are involved in those regions. So what we found was that in those regions, they were mostly regions of loss in the blue color. They were loss of tumor suppressor genes, and in the same sample, there were loss of oncogenes. So by inference, we're postulating that these actually creates like a balancing effect and protects from transformation into melanoma. This, of course, has to be proven in a much more intricate study. So we think that these balance events are really, really common, and that leads to benign nevi, and that's the primary event. And if there were imbalanced events occurs, then this leads to these intermediate lesions and leads on to melanoma in situ. So just in the interest of time, I'll move on as quickly as I possibly can. We've got less than 10 minutes to go. What we also found was that in these nevi on the sun-shielded body sites, there was a predominance of these insertion deletion type mutations. So we wanted to explore that a little bit further. So I presented this work at a conference a few years ago in Manchester, and after the presentation, I was approached by Rajiv and Eduardo, and so they were really interested in the work, and they said that they've got some exome data already on nevi, and so they wanted to collaborate. So I was very happy to do that, of course. So their nevi were primarily from sun-shielded body sites or minimally exposed body sites. So we wrote this up and published this a few months ago. And what we found was that between the Spanish cohort from Eduardo and our Australian cohort, that the profile was quite different. The Australian cohort is in the blue here, so it indicates that there are different origins. So in a nutshell, just very briefly, our sun-exposed nevi have a much higher UV signature. Minimal sun exposure have much lower ones, and they primarily have these insertion deletion mutations. And when there was no UV signature, there was more dominantly this mismatch repair signature, this defects in mismatch repair. So we'll talk to you briefly about that shortly. So very quickly, to finish up on these large genital acquired nevi, not acquired nevi, where they're born, people are born with these entities. So this paper has just been accepted a few days ago in JID. So this is in collaboration with our Spanish collaborators in Barcelona. So just very briefly, these are clearly present at birth. They range in size from small to giant. So we were focusing on these giant nevi, which are very heterogeneous in their appearance. They have an increased risk for melanoma transformation, and they commonly have this post-zygotic mutation in NRAS. So this is the paper that was published 2 years ago by our collaborators in Barcelona. So we really wanted to follow up to see what else was there. They only performed targeted mutation detection, not whole exome. So it was really important to see what else was happening. What they found was in essence that 60% of these these congenital nevi have a MAP kinase mutation in NRAS. So what we wanted to do was look in these samples more closely. So we sought to investigate if there's actually anything novel, any novel mutations alongside NRAS, but also to discover anything that wasn't present that was undetectable in NRAS wild type samples. So we performed the same type of analysis as the acquired nevi. We performed whole exome sequencing, really deep sequencing to make sure we weren't missing anything. So the mutation rate was really low, way lower than acquired nevi, only up to 1 mutation per megabase, whereas in acquired nevi it's up to 9 mutations per megabase. The insertion deletions were 2 times higher than the single nucleotide variants, and this was actually consistent with our study of the acquired nevi located in body sites with minimal sun exposure. So we confirmed the presence of the NRAS mutation, but in those patients with the NRAS mutation, there were actually novel mutations present at the same mutation frequency, which indicates that they also contribute to the formation of the nevus. In the NRAS wild-type patients, this is an example here that we had a biopsy taken from the shoulder and a biopsy taken from the satellite lesion. There was a novel mutation in the lunatic fringe gene. So this is a member of the Notch signaling pathway, and it's actually crucial for embryonic development. So we don't know exactly what it is doing in congenital nevi, but we do know from previous papers that it's involved in the Notch P10 AKT pathway. This suggests that there's an alternative pathway to development of these types of nevi. Mutation signature analysis confirms that there are definitely no UV signatures, which confirms that these are congenital in origin, but there was very common mismatch repair signatures in almost all of the patients. So this helps explain these insertion deletion mutations. There were, however, no somatic mutations in the pathway, but that's just of the known genes. There's a lot more work to be done, obviously. So we think that mismatch repair defects may play a role in the development of the nevus. So just to finish up, you looking at copy number, you saw this before. In the giant congenital nevi, the copy number is very, very different. So they're much more small, discrete changes compared to the acquired nevi. And in summary, hopefully I've got the point across that common acquired nevi are 100% mutated for the common BRF and NRAS mutations. Congenital nevi are 60% post-zygotic NRAS. Exome sequencing discovers novel initiating mutations. Nevi from these minimal sun-exposed body sites are very similar profile to congenital nevi. Mismatch repair is common when UV is absent, and then copy number aberrations are absent or balanced in nevi. So lastly, obviously, just to thank, there's a whole lot of people involved in here, and particularly my funding partners. Thank you so much.
Thank you very much, Mitch. That was terrific. And thank you again to Rick for his talk previously. So we're a bit short of time, but we've probably got time for a couple of questions. Nicholas, you've posted one. I think that's an interesting question. Do you want to phrase it for Mitch?
Yeah. So yeah, very nice talk, Mitch, and Rick as well, of course. So I was wondering, and I see this quite often in the literature, why do we compare nevocytes to the adjacent normal skin? Because the only related the only related cell type in the normal skin is the melanocyte, which is actually relatively rare. So aren't you contaminating your comparison with non-related cell types like keratinocytes, fibroblasts, and all kinds of other cell types that are in the skin when you do these comparisons?
So the main reason we're looking at the skin, Nick, is because the nevi itself also has keratinocytes in the tissue. So we didn't perform any microdissection or macrodissection. We used the whole tissue to see what the whole spectrum was and what the contribution of the keratinocytic mutations were. Yeah.
Would you expect a difference if we only compare nevocytes, meaning the melanocytic component, to melanocytes?
To the melanocytes of the skin? Of
the unaffected adjacent skin, yeah.
That's a good question. I'm not quite sure. We have Rick's team has cultured the melanocytes from the skin and cultured the melanocytes from the nevi, and the profiles that we're seeing are almost identical to the whole tissue. So the same types of signatures are there. So I think what we're mainly seeing actually is coming from the melanocytes,
right?
Because it's the clonal entity rather than the keratinocytes, which are slothed off quite regularly.
Great, thanks.
Yeah.
So Mitch, if I understand rightly what you're saying the melanocytes from normal skin have the same UV signature?
Only if there's enough of them to form some sort of clonal entity to detect those types of signatures.
So these aren't the normal melanocytes you're talking about, Mitch. These are the nevocytes that have been exposed to UV for 30, 40, 50 years.
Well, that's true, Rick, but from the cultured the cells that you have, the patients that have had chronic UV exposure, which you can tell from their clinical photos as well, they are the only ones that have UV signature in their skin, in their melanocytes cultured in the skin. Whereas people that have hardly any UV exposure, they've got nothing. But the nevus does. Yeah. That makes sense. It's very, very, very strange.
Okay. Further questions?
Which I was interested in the comparison with the Spanish data. So could you expand a little bit more on that, given what you just said about the UV signatures?
Yeah, the nevi themselves, they had the same dermoscopic patterns that we observe in our Australian cohort, but I'm almost certain that these were not acquired, as in from sun-induced. So Peter can talk more about exactly what this means, but there's an entity called tardive congenital nevi, which they're not present at birth per se, but they're present under the skin, and they have risen to the surface over time. And I think that's why they look just like the giant acquired congenital nevi.
Yeah, and I'd like to confirm this. The nevi, which we clinically often call acquired, are probably tardive congenital nevi.
Yeah. So I think that's what we were studying in that Spanish cohort, and they're mainly excised. I think those ones were excised in that particular study just for cosmetic reasons. They weren't anything wrong with them. The patients just wanted to have them taken off, but they happened to be in body sites that had hardly any sun exposure.
Yeah, I mean, because in general, the work is so exciting because there is this puzzle still. Okay, some of the nevi obviously then turn into melanoma, and I think you've explained well why that might happen, because there's another hit. But then all the others that arise from we don't know exactly where, that's sort of the interesting question that you have to solve.
Yes, we're still looking very heavily at different sun-exposed areas of the skin to try and find the same types of patterns that we're observing in nevi. I mean, would that indicate that area is more likely to develop melanoma over another area? There's a lot more to do, way more to do.
Yeah.
So is there any information on comparing British people to Australians? Because Spanish people obviously are dark-haired, dark-eyed. They sleep from 12 to 3 p.m. Australians go to the beach at that time. So is there any comparison to British people, which would be the same, more or less the
same
genotype then?
Yeah, I've been talking with Julie Newton-Bishop about collecting tissues from UK individuals, but that COVID obviously got in the way, so that's still on the cards. Yeah.
Rick, can I ask you a question, a sort of naive and very general question? What's known now about epigenetic contributions, if any, to melanoma?
It's a good question. Certainly the epigenetic modifiers, the histone tail modifiers, are very important in melanoma and often mutated. I should really write a review on that subject. Yeah.
All right. It's a good question, right? But we don't quite know what the answer is yet.
Yes.
Yeah. Okay. All right. Look, we're a little bit over time, but look, can I thank you both for really excellent talks on behalf of everybody? I thought that was really fantastic. Rick, I enjoyed your reminiscences, and especially the history of your facial hair was really extraordinary. No, it was really, really fun. I really enjoyed that. Thank you so much, both of you.